Good morning to everybody. First of all, I want to thank the organizer of this workshop for inviting me to give my talk. Today I'm going to present some results of a multidisciplinary study that involved the collaboration of psychophysiologists, bioengineers, and also biochemists. The object of this research that was funded by the University of Pisa is the brain-body interplay, brain-body interaction during stress-related, emotional-related conditions. The causal relationship between information that comes from the body and brain activity during the emotional experience is a long-lasting, is the subject of a long-lasting debate that goes back to the dispute between the theories of James and Cannon. This debate is ancient, but is still open. However, nowadays, there is a general consensus in considering the primary role played by the complex brain-body interaction in giving rise to conscious experience and thus also to the emotional experience. Si sente? Okay. Specifically, in our study, we investigated the bidirectional brain-heart interaction during a condition of test anxiety and also its possible modulation by female ovarian hormones. A few words about bidirectional brain-heart interplay. Both brain activity and heartbeat activity show very complex oscillatory patterns. When I say complex, I mean they are time variant, frequency dependent, and follow nonlinear fluctuations. These activity patterns are likely coordinated with each other through complex causal interactions that are likely modulated by the sympathetic and parasympathetic branches of the autonomic nervous system. This dynamic information interplay between the central autonomic control networks and the peripheral autonomic nervous system is termed functional brain-heart interplay. Many methods have been proposed to study this bidirectional brain-heart interplay, specifically to infer about the directionality of this parallel oscillation. Here, in this study, we implemented the synthetic data generation model developed by the group of Professor Valenza that evaluates the bidirectional modulation between EEG, electroencephalographic oscillation at a given frequency band and spectral estimator of sympathovagal activity extracted from heart rate variability. In particular, we used the low frequency and high frequency spectral estimators of sympathovagal activity. HF is considered a good estimator of parasympathetic vagal activity, while LF is not a pure sympathetic estimator because contains both sympathetic and parasympathetic information, but is considered a good estimator of baroceptive activity. According to the SDG model, time-varying directional biomarkers of brain-heart interplay represent an immediate assessment of brain-to-heart, heart-to-brain interaction for different combination of EEG frequency band and the LF-HF bands of heart rate variability. A few words about test anxiety. What is test anxiety? Is a type of performance anxiety, is the most prominent cause of anxiety among students. It's a multidimensional construct defined as a set of physiological, cognitive, emotional, behavioral responses that accompany worries, concern about a possible failure at an examination. Here is a long list of typical symptoms that accompany test anxiety. These are a list of physical, behavioral, cognitive, affective symptoms that goes from heart rate increase, sweating, dry mouth, but avoidance behavior, but also cognitive, like going black or fear of being assessed. In general, we can say that a primary indicator of test anxiety is the increased level of physiological arousal. Just a few words about brain modulation by ovarian hormones. Hormonal information is conveyed to the brain via the circulatory system. So hormonal information belongs to the a non-neuronal interoceptive pathway, is the so-called humoral interoceptive pathway. And what is interesting is that evidence, especially from the animal models, have shown that many regions, many brain regions involved in emotional processing, but also belonging to the central autonomic networks, have many receptors for ovarian hormones. For instance, amygdala have a receptor for both progesterone and estrogens. Also many hypothalamic regions, but also NTS, that is an important center of autonomic regulation, have many receptors for estrogens. Also from there are many evidence indicated that hormonal fluctuation during the menstrual cycle modulate responses to emotional stimuli, especially stimulations that have a negative valence, but also to reward. Okay, in our study, we used as a test anxiety task a simulation of an oral exam, and our participants were 24 female healthy students. Our aims was to describe the bidirectional brain-heart interplay during the simulation of the exam and to investigate a possible ovarian hormones, the modulation of ovarian hormones on this brain-heart interplay. The experimental protocol consists of an initially relaxation phase, followed by the test anxiety task that consisted of 3 successive phases: base, study, or representation. During the study phase, the participant had to read and study a brief test presented on the computer screen, and then the subject had to make a short presentation in front of a professor, that in that case it was me, that was supposed to evaluate and assign a score to the performance. Throughout across the whole session, we recorded the EEG and the electrocardiogram by using the G-Nautilus 32 multipurpose wireless system that allow the synchronized recordings of EEG and ECG signals. We also collected salivary samples to measure the hormonal levels, but also to investigate changes in salivary proteins that are considered good markers of performance anxiety. We also evaluated the subjective changes in the perceived anxiety by using a state anxiety scale, in particular the STAY-Y1 scale. Participants were recruited at 2 different phases of the menstrual cycle. The pre-ovulatory group was recruited at the very beginning of the menstrual cycles, that is characterized by low level of ovarian hormones. The other group, the post-ov group, was recruited at around the 20th day of the menstrual cycle, that is characterized by medium levels of estradiol and high levels of progesterone. We then computed by the EEG-ECG signals, we computed the brain-heart interplay indices for different combinations of EEG frequency band. In particular, we used the delta, theta, alpha, and beta EEG bands, and LF and HF indices of heart rate variability. These indices were calculated for the brain-to-heart, heart-to-brain interplay at the individual EEG electrodes for the 3 phases of the experimental session. Okay, here are the main results. The first result I'll show you refer to the analysis of the whole sample, not subdivided in these 2 subgroups. First of all, we wanted to, in order to detect a possible difference in brain-heart measures across the 3 phases, we used a repeated measure analysis. In particular, we used the Friedman test. And here is the topographical representation of the group-wise comparison for the brain-to-heart interplay. As you can see, in red are the significant differences here. We found significant differences for the brain-to-LF interplay, which involve almost the entire scalp, and these differences were found for all the considered EEG frequency bands. As for the heart-to-brain interplay, we found differences for both LF to brain interplay, but also to HF to brain interplay. In particular, we found significant differences in the posterior central region for delta, theta, and the alpha band, but also for the frontal region in the delta for delta. As for HF to brain interplay, significant differences were found in bilateral frontal regions for delta, in the right frontal region for theta, and in the central posterior areas for alpha. We then performed pairwise comparison between BHI indices of the different conditions by using the Wilcoxon test. Here are the topographic representation of the significant results. As for the brain-to-heart interplay, we found that significant differences were mainly associated to the presentation phase, which show lower brain to LF measures with respect to both the basal and the study phase. We found, as for the heart-to-brain interplay, differences were mainly related to the study phase, which appears to have a lower LF to brain and the HF to brain measures with respect to the basal condition, but differences were found also between the S phase and the presentation phase for LF to theta and LF to delta and LF to theta measures. We then compared the two subgroups, the pre-ovulatory and the post-ovulatory groups. So we compared the BHI measures of the pre-ov and post-ov group by means of Mann-Whitney. Here is a topographical representation of this comparison, and the statistical analysis yielded significant difference during the study phase. In particular, during the study phase, the post-ov group showed higher brain to HF interplay for all the considered EEG frequency band, and these differences were found in the central region of the scalp. We also studied possible association between BHI measures and the hormone levels, and we used Pearson correlation, and Pearson correlation analysis revealed significant association between brain to HF measures and the progesterone levels only in the pre-ovulatory group. And these differences were found in the midline right frontal regions and at central areas. Okay, in conclusion, test anxiety induces fluctuation in brain-heart interplay, which mainly involve interplay between brain and the LF heart rate variability spectral component. LF heart rate variability spectral component is considered a good estimator of baroreceptive activity. Indeed, this finding is not surprising because from the literature we know that there are indications, there are evidence of a modulation by baroreceptive activity of both emotional but also cognitive processes, for example, memorization of a list of words. Our results are also in line with the previous research that indicate a primary role of this dynamic bidirectional brain-heart interplay in both sustaining but also initiating the emotional arousal and mental stress. Okay. I think the most important results is the finding that the brain-to-heart interplay may be modulated by different levels of the ovarian hormones. In particular, we found a modulation that involved the brain to heart, in particular the interplay that involved the vagal HF activity. the non-neuronal hormonal modulation of brain-heart interplay during anxiety-related condition is, to our knowledge, a novel finding. Our future perspective, first of all, to increase the sample size, and then we are in this moment, we are actually involved in completing the description of the salivary protein changes. Here is an example. Here is a two-dimensional electrophoretic plot. Each spot, different protein, and most of them have been recognized, but not all these proteins are known. And we circled the areas which showed changes. The protein changes, the protein expression changes changed after the test anxiety. So what we are thinking to do in the near future is to try to find a possible association between the levels of higher hormones, the salivary proteome, but also BHI measure, and also the subjective scores of perceived anxiety. Thank you for your attention.
A question?
Hello, thank you for this complex series of correlations between many variables. I have two questions. One question is, when you record EEG in these three different phases, subjects are doing very different things, right? Because in the first phase, they are just at rest. In the second phase, they are assessed by you, if I understand. And in the third phase, they have they produce an oral presentation.
It's completely different. The first phase, they just are doing nothing, just staying with open eyes and watching at the wall in front of them. The second phase, they're studying. So it is a phase where there is a high cognitive involvement, attention, working memory, and stuff like that. And the last one, I think it was the most emotional phase because they have to do the oral presentation.
Okay. Okay. So one question just to understand is whether you're sure that the differences that you see in brain-heart interaction during these three phases are not due just to the different brain activation related to these three phases.
Okay.
And the second question is, you showed us the novel result, this interaction of the hormones with this brain-heart interaction. Can you just tell us an intuition about what really happens, what you are discovering functionally?
Okay, this is they are both difficult answer to reply because it's a problem when you use a task. When we do experiment, we can use a very simple experiment where you can control all the aspect. Here we use a more ecological is the term, okay, condition. So it's a real simulation of an exam. So all these factors are involved, and we have to think how to try to separate this the different variables, because we know that. For instance, I did not tell you, but for example, the language network is always involved. Okay, they have to read and then you have to talk. Okay, so this is a big problem. But we think that anxiety is a common aspect that is present throughout the whole session. So we think that we could, by analyzing the brain-to-heart or heart-to-brain interplay and the different EEG oscillation involved and the different component of heart rate variability involved, we could try to discriminate the different aspects. The second question, the hormones. I was personally interested in these two aspects because there are two different ways of interacting with the brain. The body interacts with the brain. One is through the neuronal mechanism. The other one is a neuronal but very pervasive way to act on the brain. And also the other aspect that I'm interested in is that these brain levels, these hormonal levels are oscillating. This could be with a different time course with the neural activity. So this could be another interesting aspect to develop and to study.
Okay. We thank again Laura.